Thanks so much for inviting me to this very prestigious lectureship. I was thinking, when I got invited, I wanted to come up with a talk that would really impress you. And then I thought, what a ridiculous idea. Here I am in a city that has had, lectures describing the most important discoveries of mankind over the last century or so. And I thought, I'm not going to say anything that's going to impress you. So in thinking about that, I thought, well, what could I do? And I thought, actually, I think I'm just going to tell you a story about what I've been interested in. And then you can decide, whether that's an interesting story or not. And as Ron was saying, I wanted to start off with where I started at the NIH and what seemed really interesting to me at the time, and where that sort of led me in my career at the NIH, and how I was thinking about, as I'll show you, in the early days, I was really intrigued by the concept of receptors and how they caused cells to change their behavior. And in the course of that, as Ron alluded to, I'll talk a little bit about, and then I'll explain how that, a very basic idea, led to a new class of drugs, but equally how interrogating patients leads you backwards to think about these things again, and then the other thing that happened, of course, during the duration of doing these experiments is, we completed the human genome for one, and now we have all sorts of genomic tools that you guys just take for granted, but frankly, they're things that we couldn't even conceive of when I started thinking about this. So I think, as you'll see, we filled in a lot about this black box over the last 25 years or so, but I think equally you'll see that we're just at the beginning of understanding these processes. So when I was an undergraduate, I took a course, and it was about the time when this paper came out, and they had made this spectacular discovery of how receptors sat in the plasma membrane, and they were these little, this was like, potatoes in this soup, but importantly, what they did recognize was, how this might happen physical chemically. At the same time, as intriguing as this was, it sure didn't tell you a lot about how a signal out here could change a cell's behavior. So I came to the NIH, as you heard, clinically trained, and as the students, we talked about at lunch, that I really did get the bug for understanding basic processes, and as you heard, mostly because I had been a clinician, and I thought about options, and doing research seemed way more interesting than some of the other options. And really, the NIH was the right place to go for a young clinician. But I did realize right away I was at a disadvantage because I didn't have a PhD. And so I knew medicine, but there was a whole lot that I didn't know. And I certainly didn't know much about this process of receptors and receptor-mediated signal transduction. But I was fortunate enough to take a course at the NIH in biochemistry and signal transduction, which ironically I now teach in. But this was one of the professors who was teaching this course. And he gave this amazing lecture on G-protein coupled signaling. And I remember at the time I was just spellbound. I thought, wow, I guess I sort of had been sleeping during that part of the class in medical school, or maybe they hadn't discovered it yet, probably more the latter. And I remember when I went up to him and I said, that was a great talk, And he said, thank you. And then shortly thereafter, he did win the Nobel Prize. So, he was probably really waiting for me to thank him or to say how great a talk it was. But I was thinking about this when I was putting my talk together, and I thought, oh, that's really so exciting now to be coming back now, and then to, again, be thinking about how this problem, I guess he got the Nobel Prize in about 1995 or so, and just think about how this problem has changed since I heard these lectures in the 1980s. And so what was happening at that time is that, of course, this was the molecular biology revolution. And so we went from thinking about these, potatoes swimming in this lipid soup to really having structural insights into receptors. And so, again, I remember very vividly at one of the meetings I went to when the insulin receptor was first cloned. And the exciting discovery was that the insulin receptor was a receptor tyrosine kinase. So for all the, sort of struggling of trying to understand how such a receptor could work, suddenly that was easy. The receptor is a kinase. And you could imagine if insulin binds, it activates the enzymatic activity, and you induce phosphorylation of substrates. So that was tremendously exciting. What was also exciting, of course, was when you added cytokines to cells. That also induced tyrosine phosphorylation. But actually, the cloning of of cytokine receptors was a little bit of a disappointment. One of the first cytokine receptors that was cloned, my colleague Warren Leonard at the NIH cloned the alpha chain of the IL-2 receptor. And again, that doesn't even have this configuration. It really has a very small nubbin of a cytoplasmic domain. So suddenly, that answered a question, but it raised many other questions. And really, the question was what's the kinase that's responsible for signaling via cytokines in this class of receptors? So it was solved, as you well know, by the discovery of the JAK-STAT pathway by Darnell and George Stark and Ian Kerr and others. And it came from two approaches, really. One was just screening libraries for kinases, but then also a pivotal experiment by Sandra Pellegrini, who showed that if you took cells that weren't responding to a cytokine, in this case interferons, and reconstituted them genetically, you restored the pathway. So that really put JAKs, in this case it was just to make things complicated for you, the JAK was called TYK2, but as I'll show you in a moment, TYK2 is a Janus family kinase. But that restored signaling by interferons. And Jim Darnell, of course, took a more sort of brute force approach by identifying complexes bound to interferon-induced genes, discovered STATs, of which we now, of course, have 7 STATs. But what we now know, of course, is that this is a pathway that's employed by more than 60 cytokines and growth factors. It's evolutionarily conserved, really easily from flies to mammals, where the pathway looks very similar. Dictyostelium to mammals also have elements of this pathway, and I think dictyostelium, remarkably, has 4 STATs. So this is a pathway, right, when you think of dictyostelium as this organism that can't quite decide whether it's a unicellular organism or a multicellular organism, and it sort of changes when if things get bad, they decide to band together. And so this obviously is a very, very ancient pathway that controls an organism's response to the environment. And what we know now, of course, is that this pathway is critically important for development, growth, hematopoiesis, metabolism, and immunity. The question, of course, is that what hadn't been sorted out really was the in vivo criticality. So in these cell lines, mutagenized cell lines, we knew that this pathway was important. But again, this was at an era where people then moved from experiments in cell lines and this sort of thing to looking in vivo and using knockouts. Now, in this case, the first knockout in the JAK-STAT pathway didn't really come from a mouse. It came from studying humans. So Gigi Notarangelo and Rebecca Buckley had patients who had a very particular type of immunodeficiency. And what was discovered, again, my colleague Warren Leonard at the NIH had discovered that mutations of the common gamma chain, a receptor that's used by IL-2, 4, 7, 9, 15, and 21, was lacking in boys that had this particular type of immunodeficiency where they had no T cells, they had B cells, they had no NK cells, referred to as T minus B plus NK minus SCID. When we cloned JAK3, we had shown that it's activated by things like IL-2. And then with Warren, I was able to show that JAK3 and the common gamma chain were associated. And when this paper came out, people like Gigi Notarangelo and Rebecca Buckley reasoned appropriately that, that maybe there were girls who had T minus B plus SCID who had not mutations. They didn't have mutations in the common gamma chain, of course, because it's XSCID. But autosomal recessive SCID might be due to a JAK3 mutation. So we collaborated with Gigi and Becky Buckley and in fact, reported girls and boys who had SCID due to JAK3 mutations. So that was important for a couple of reasons. It demonstrated that, in fact, JAK3 is critical for signaling via the common gamma chain. But it also was important because this disease really phenocopied this disease. So it really said that the function of this kinase was for signaling via this receptor. It didn't have broader functions. This mutations, this receptor didn't have broader functions, that this really was the pathway. So around this time, of course, there was a lot of interest in targeting cytokine receptors. Again, this was the era, molecular biology explosion, where many receptors and many cytokines, et cetera, were identified. And then, of course, we began to make monoclonal antibodies against cytokine receptors. So when we published this paper, we made the bold claim way back in, whatever it was, 1995, that if you targeted JAKs, that would represent a new class of immunosuppressive molecules, immunomodulatory molecules. So about this time, I was at a meeting in Saxton Rivers, Vermont, and just presenting the cloning of JAK3, and I ran into a guy, Paul Chingelian, who had actually, I knew previously we'd both worked on complement prior to what I was working on now, what Paul was working on, and I said, Paul, what are you doing at this meeting on, I think it was antibodies, and it was an immunology meeting. I said, what are you doing at this meeting? And he said, oh, I've now moved to Pfizer, and we're looking for tyrosine kinases to target. And I said, wow, I got a winner for you. And so, in fact, through a collaboration then with Paul and a chemist, Mark Flanagan, Pfizer generated this compound here called Tofacitinib. And this was an example where at the NIH, we certainly can collaborate with industry. And this was done in the form of, it's called a CRADA, which stands for something, I don't know, Collaborative Research Agreement and Development Award, right? And so really we've had, since that time for probably a couple decades, we've had a collaboration with Pfizer. And again, for me, it's been very, very exciting because you sort of realize right away the strengths of academicians, and then you also recognize very well the strengths of industry in really taking, it's just sort of an offhand comment in a discussion section of a paper to moving something like that to reality. So this is the current status of JAK inhibitors. There are the FDA-approved JAK inhibitors shown here. Tofacitinib I'll come to in a moment. Ruxolitinib was actually approved by the FDA first. And again, that was sort of a little bit of a funny story that ruxolitinib is a JAK1, JAK2 inhibitor. It was recognized during the interim that the disease polycythemia vera and myelofibrosis is due to gain-of-function mutations of JAK2. And so, in fact, Incyte was well poised then to develop and study in clinical trials their inhibitor in this disorder. It's being tested in other disorders as well. So I'm going to come back to this again. I'll simply point out now that at around this time, people then made knockouts of all these different mice. And if you made a knockout of JAK2, what you ended up with embryonic lethality. JAK2 is the enzyme that's important for signaling via erythropoietin. So you can imagine, as an embryo, if you can't make blood, that's not a good thing. So, and I'll come back to that. So this whole idea in drug development of using genetic tools and predicting utility in clinical scenarios from your genetic tools, et cetera. This I think is one of the sort of interesting things to sort of to think about, and JAK inhibitors are a particularly useful lesson in this regard. I'm going to come back to that. Tofacitinib, again, we had a CRADA with Pfizer. This drug is a JAK1, JAK3 inhibitor, and I'll tell you more about that in a moment, and less a JAK2 inhibitor. It's approved in the United States for rheumatoid arthritis, and there are ongoing trials in juvenile arthritis, spondyloarthropathies, IBD, and psoriasis. So I was telling Ron, and we were at dinner last night, and I was in Amsterdam I guess about a year or so ago. And again, they invited me to come speak about JAK inhibitors. And again, they were very, very gracious, and they referred to me as the godfather of this class of drugs and everything. Like, there's very flattering. But at dinner, somebody told me a story that's very similar to the history that I'm going to tell you. So I'm going to tell you about Leo. So Leo is a 6-year-old, and he was well until about 3 years ago when he developed severe dermatitis. And his mother brought him in to see the doc, and he was diagnosed as having food allergies, and then they did everything you do with food allergies. You restrict the diet and all this complicated stuff. And he wasn't getting better. And then he was put on steroids, and again, his mother was really, really worried, putting a 6-year-old on steroids, what's that, all the side effects of steroids and everything. And still, he wasn't doing well. And this is Leo right here. So you can see Leo is just miserable. You can see all the dermatitis. Leo was very unhappy and really had a modest response to prednisone. Then his mother found out about JAK-inhibs, and Leo was put on JAK-inhibs. And you can see Leo is a happy dog. And Leo's mother works at the NIH. Leo's mother is a scientist. And ordinarily, if you've seen me give this talk, I think I usually black out the dog's eyes for the privacy purposes. But Leo's mom said very specifically I could use these slides, and was actually thrilled that Leo is going to be shown at the Nobel Forum, and Leo was wagging his tail when we discussed this possibility. So that leads to the third FDA-approved JAK inhibitor, Tofacitinib, which actually came from the original screen of Pfizer. Pfizer then spun up, this is a different company now, but it was originally part of Pfizer. And Tofacitinib is a JAK1, JAK2, JAK3 inhibitor, and it's approved for dermatitis in dogs. And I can tell you, often when I go around and give this talk, at the end in the reception, I often have more people asking me about their dog than patients with all these diseases. I should add, though, I think it's actually very interesting having this veterinary experience, because now we have many not people, but many subjects, if you will, on a JAK inhibitor. So we're really rapidly expanding what these drugs can do. And it was actually interesting, I won't go into the details now, but how these trials were done to get FDA approval. Now, in the meanwhile, people are using JAK inhibitors for other things. You could see that one thing that really attracted everyone's attention is that JAK inhibitors can cure baldness. Needless to say, I got very excited about this. But that this is a particular type of baldness, and this is autoimmune-mediated baldness, several types in which JAK inhibitors have been used. And the drugs have also been used for other immune-mediated diseases, such as vitiligo. So let me just go back and just imagine, if you will, when I sort of first went to Pfizer in, the mid-90s, and made the pitch saying, oh, no, you guys should target JAKs, and what we were thinking. So at the time, of course, based on our discoveries of the pathway and then the patients, we thought, well, what you really want to do is make a selective JAK3 inhibitor. Now, again, without going off on too much of a tangent, I would just sort of remind you, back then, before we had so many tyrosine kinase inhibitors, believe it or not, it was actually controversial as to whether tyrosine kinases would be a good idea or not. You might imagine that if I said, well, I think it's a really good idea to target an enzyme that uses ATP, that people would look at you and think, really? How are you going to get some specificity? Now, we all take that for granted now, that chemists are so good that they can make selective inhibitors. But the idea then of targeting an enzyme that used ATP seemed ludicrous. But if you said, okay, well, what we really need to do is target this particular enzyme, it was a little controversial, but people were okay with that. Now, I point out here that at the time we thought of IL-2 as targeting, JAK3 as being important for IL-2, 4, 7, 9, and 15. And again, based on the patients, we were thinking of primarily this drug is targeting adaptive immunity. But, and this is another point that I'd like to sort of make, is that when a drug development project starts, you have a certain batch of knowledge. And then what happens? Well, then you go ahead, and often it takes 20 years to get a drug, and science doesn't stand still. It changes, right? So you think about way back in the mid-90s, we didn't know about IL-21, right? We didn't know about FOXP3. We didn't really think about Tregs. Th17 cells hadn't been discovered. And Charlie Janeway, actually the first time I sort of learned about the concept of innate immunity was actually here in Stockholm. Charlie Janeway was at a talk, and he was pitching this idea of innate immunity. And I remember thinking, what is he talking about? What is this innate immunity thing? This is, if you're working on, this is all non-specific stuff. This is really where all the action is. And so we didn't really even think about how this drug might be targeting things other than JAK3. That seemed very logical. And then on top of that, well, the human genome hadn't been completed, so we didn't even use the word kinome. So you didn't have the opportunity to think about selectivity on a broad scale. But now, of course, we have the human kinome. There are 518 kinases, 90 tyrosine kinases, and 4 JAKs. Again, when we were just cloning the JAKs, we didn't really know how many JAKs we would ultimately have. So now it's a pretty straightforward thing. If a company says, you know, I have a new inhibitor, a new JAK inhibitor, as a reviewer, you just say, oh yeah, you know, show me, buddy. And this is the experiment that they can readily do. They can take the kinome and then say, okay, does the drug have some kind of selectivity for the kinase you want it to have? You say it has actions against, but more importantly, perhaps, does it have activity against other kinases in the kinome? So that was done with Tofacitinib, not by Pfizer, but by other companies. And we now know that Tofacitinib, for example, the big circles means high activity towards that kinase. For JAK1, and not shown here, JAK2 and JAK3, in vivo, it has a little less selectivity it has less activity towards JAK2. And pretty much not a lot of activity towards anything else. Another enzyme down here that it's not clear that has any relation to efficacy. But so now we know that JAK3, Tofacitinib inhibits JAK3, but also inhibits JAK1, and then to a lesser extent JAK2. And I could only imagine if I had gone to Pfizer, you know, back in the early 90s, and I said, you know what, why don't you just make an inhibitor that activates JAK3, JAK1, JAK2, and that'll be fine. That'll be fine if you're inhibiting all these JAKs. They would have said, thank you very much. Don't come back. That's the stupidest thing I've ever heard of. But as you see, we're in a little different era now. And in fact, these are all the cytokines that are potentially targeted by a drug that inhibits multiple JAKs. So Tofacitinib will inhibit JAK3, JAK1, and JAK2. And so you're inhibiting the thing we expected to inhibit, adaptive immunity, but you're also inhibiting cytokines that are really pivotal in innate responses, like IL-6 shown here, also to an extent, IL-12, IL-23, interferons, et cetera, GM-CSF. So it really does raise the question, when you see efficacy in a patient, or if you see efficacy in Leo, which are the cytokines that what is the basis of the efficacy that's making Leo a happy dog versus our initial views of it inhibiting strictly adaptive immunity. So in fact, the first generation JAK inhibitors really have broad effects on innate and adaptive immunity. And it really makes you wonder, of all these different cytokines that are being targeted, really what's the basis of efficacy? And then going forward, of course, you think, okay, how can we improve that? And that brings the issue to next generation JAK inhibitors. And so now there's good evidence that drugs like Tofacitinib, which target multiple JAKs and therefore multiple cytokines, have efficacy. And now that's joined by other drugs like baricitinib that target JAK1 and JAK2. And again, this drug also has efficacy as shown in phase 3 clinical trials in rheumatoid arthritis. But now added to that, drugs are being developed in which you target a more restricted panel of JAKs. Filgotinib is a drug made by Galapagos in Belgium. This is an AbbVie drug, I think, that targets JAK1, so you could see that it targets a narrower spectrum of cytokines. And Dasatinib is a JAK3 selective inhibitor in which the phase 2A clinical trials were published, and now the phase 2B clinical trials are now online, or soon. So this is, to me, this is a really exciting time in the history of JAK inhibitors as to, you see all these drugs that have efficacy, how are they working, where are they working, et cetera, et cetera. Now, it's a little, it's a critical question, again, as people are saying, well, this is a JAK1 inhibitor, this is a JAK3 inhibitor. Of course, like I said before, you'd say, okay, I want really you to prove that to me in an in vitro assay with a recombinant enzyme in a cell, but most of all in people. And this is sort of the latest scorecard of JAK inhibitors, including the 3 FDA-approved inhibitors, ruxolitinib, tofacitinib, the canine JAK inhibitor, baricitinib in late-phase clinical trials, filgotinib, and I don't think I have the ABT compound here, but suffice it to say, there are many JAK inhibitors that are being developed, and we may very well see in broad clinical use soon. But I just wanted to take a few seconds and think about, okay, where did we think things were in 1995, and how did it actually pan out? So when we made our bold claim in the Science article in 1995, we predicted the efficacy. That seemed like an obvious thing to predict. We obviously had no idea of the safety because the drugs were inhibiting cytokines that are important for host defense. Now, at the time, if anybody had asked me, did I think selectivity was essential, and I think everyone in the field would have said selectivity would be critical, assuming you could get it. So that's changed a lot in the last decade or a couple of decades. That again, chemists can seemingly easily do this. I mean, I couldn't do it, but chemists in pharmaceutical firms are very good at this. But I think no one would have predicted that a pan-JAK inhibitor would be viable, and so a drug like baricitinib, which inhibits JAK1 and JAK2, maybe I'll just go back for 1 second. So if baricitinib inhibits JAK1, JAK2, and really just recall that this family of cytokines, 2, 4, 7, 9, 15, and 21, are also JAK1 dependent. So if you had a JAK1 and JAK2 inhibitor, in principle, it should do pretty much the same thing as a JAK3, JAK1 inhibitor. But you would be a bit worried, based, as I was saying, that the knockout of JAK2 was embryonically lethal. Knockout of JAK1 is perinatally lethal. So I don't think that anyone would have predicted a vice president at a pharmaceutical firm would be particularly excited if their group came to them and said, oh, well, we got an inhibitor, pretty much inhibits all the JAKs, but don't worry about it, it'll be a fine drug. So, and in particular, I think people would have been very concerned, and companies were concerned when they had JAK2 inhibitors because of the lethality of the knockout mice. But I think the answer now is that, well, a drug doesn't make you a knockout. A drug, you give it in doses. And so on one level, the genetics really help you think about what a drug can do, but in fact, they don't actually predict what's going to be useful in people or not. I certainly predicted back in those early days that JAK inhibitors would be useful for diseases mediated predominantly by adaptive immune mechanisms. But as I alluded to, I never really even thought about innate mediated disease. I mean, even though the Charlie Janeway was telling me about this in this meeting in Stockholm that I was telling you about. But again, I wouldn't have thought that it would be such a great idea to be targeting a lot of cytokines. you would have predicted back then that one of the side effects of JAK inhibitors would be infection. That would be, you expect. If you had a JAK2 inhibitor, you would expect that you'd have anemia and cytopenia. But what you wouldn't predict was that necessarily was a side effect that could be handled. One of the other side effects of JAK inhibitors is hypolipidemia. Now again, a lot of work on that currently. Tocilizumab is also associated with hyperlipidemia, and it's a very interesting area. But again, something we didn't predict. And again, going forward, I think we don't know at this point whether a JAK selective inhibitor will be better than the first generation inhibitors or not. That's obviously needs to be tested. So again, let me just run through some things that I think are lessons from this sort of 20-year odyssey of watching this pathway evolve. I think, we still don't know the best uses for JAK inhibitors. I showed you a couple examples. I showed you Leo and his dermatitis, and I showed you some other dermatological disorders. So I think we're still learning. I think, I've thought about this before, that from the time that steroids were developed to the times where we really thought we were good at knowing how to use steroids. When I came to the NIH in the 1980s, it was 40 years or so after steroids were developed, and people were still thinking about strategies using alternate day steroids, pulse steroids, all this sort of thing. I hope it doesn't take us 40 years to figure out the right way to use JAK inhibitors. I would hope that we could speed things up a little bit, but it does really give you some pause to think that, as we'll get to, we always think we know more than we actually know. The other lesson is that rare patients are informative on multiple levels. Knockouts are informative too, but they don't necessarily predict the risk-benefit ratio for drugs in humans. Yeah, and this is a point I think I made several times. Discovering new therapies and developing new therapies takes a very long time, and that in the meantime, science doesn't stand still. And our understanding of biology changes a lot, and I gave you a few examples of that. And I think one thing that scientists often they're lacking is humility. That's not a big part of our training, is to sort of promote humility. But actually, I think we really need to embrace our ignorance and really fully understand that, just because you start here and know that the project's going to go here, that we're going to learn a ton during that time. And that's not to say you should be paralyzed by that, but you should really have that, be thinking of that. And what that does is provide opportunity for reconsidering mechanisms of action along the way. That even though our initial view is, oh, well, this is how this drug works in this disease, you're going to learn this enormous amount, and it's good to sort of keep going back. And what I would say to the graduate students and trainees is, this is great. This is job security, right? I mean, if we get all these things, if we don't understand them, that's good news for you. You'll have jobs, and you'll be able to explain to us how we were wrong and how things really work, and that's fine. Okay, let me now turn to sort of the second part of my talk, where I told you at the very beginning I was interested in this process. Is how does an extracellular signal tell a cell what to do? And we all, of course, all know from the AIDS, HIV epidemic of how important CD4 T cells are. But what we've learned in the interim is that there's all these external signals, and CD4 T cells are particularly good at sensing their environment to respond to different types of infections. And we've learned a lot about so-called signal-dependent transcription factors through the discovery of STATs, and equally we've learned about lineage-defining transcription factors, also known as master regulators, and how that drives different types of lineage commitment in CD4 T cells. A bit of a puzzle at the time was that even though all of this looked very nice and very well behaved, CD4 T cells, in fact, displayed a great deal of plasticity and had the capacity to change lineage and all these sort of things, and more importantly, also express more than one master regulator or lineage-defining transcription factor. In fact, there was fairly flexible expression of master regulators. And co-expression of master regulators, so that the cells now, somehow they were serving more than one master. So it really required us to rethink what does lineage mean, how to assess, and how to think about transcription factors. So the good news is technology came along, and one of the things that we, of course, were able to do in the early days with microarrays, but now more recently with RNA-seq, was to understand all of the transcriptome of cells that were differentiating. And so there's a lot of work on this. I'm going to tell you a very quick story on this that gave a very unexpected result. And in particular, we were interested in this cytokine, IL-27. It's an anti-inflammatory cytokine. That's its requisite in vivo function. But it was really first described as a Th1 cytokine. Its knockout was lethal when you infected the mice. And that's because of this critical anti-inflammatory function discovered by Chris Hunter. It suppresses Th17 responses and it promotes IL-10. Okay, so far so good. But here's a bit of the mystery. It uses a receptor subunit, GP130, that's also used by IL-6. So IL-6 is the prototypic pro-inflammatory cytokine, and it induces Th17 cells. It's said to promote Th2 differentiation, whereas IL-27 induces Th1 differentiation, inhibits Th17 differentiation. So we thought this was a really interesting problem to sort of think about, okay, how do we really understand specificity in cytokine signaling? So again, if you're busy and you have something to do, I'll tell you, the paper got accepted and is published, has some interesting results, but the truth is we really don't understand this. So that's the short. If you can take a short nap if you want, but I'll go through the data anyway. So what did we do? We did RNA-seq to try to understand really what does this mean, these two cytokines that were so different. How are they acting? What is the role of, they both activate STAT1 and STAT3. How does that all work? So again, by first glance, you'd say, oh, okay, look, this is a heat map, RNA-seq. You look at all of the genes that are induced by IL-27, IL-6 in red, all the genes that are repressed by IL-27, repressed by IL-6, boy, and you think, perfect. They're really exactly what we expected. They're completely opposite. Except what I'm not telling you is this. This is the whole transcriptome. All that's different is this and this. So if you look here, you'd see, well, there are similarities between IL-27 and IL-6. There are differences. But where you really see these opposite effects, that's only a very, very restricted part of the transcriptome. So that was a little disturbing to us to see exactly the ways that you had shared induction of gene expression and far more overlap than we originally anticipated. And then, of course, now we have all these really cool tools like GSEA where we can, compare things very quantitatively. And I said to Kiyoshi, who's the postdoc doing this, and I said, okay, we're just not smart enough to look through those heat maps. Use some of these great tools, and this will explain everything to you. You're going to take IL-6 and IL-27 genes, and you're going to compare them to Th1 and Th2, and versus IL-27. So again, here's Th1, Th2, and Kiyoshi showed me this, and he said, oh yeah, this is great. IL-27 gives you a Th1 response, and IL-6 gives you a Th2 response. And we said, oh, but what about this? IL-6 also gives you sort of this Th1 response. And IL-27 suppresses, or easier here, IL-6 induces a Th17 response. And IL-27 represses that, and if anything, gives you sort of a Treg response. But you see here, IL-27 also induces a bit of a Th17 response based when you have all of these genes. And so at first I said, what any good PI does to the postdoc when it comes back and says, this doesn't make any sense. Kiyoshi, go back, do it again. This can't be right. But it's right. Kiyoshi went back, and then we had, went through it all in our lab meeting. Kiyoshi was right. And so what I interpret this to mean is that cytokines are sui generis. I mean, they are what they are. I mean, if you say, IL-27 is more Th1-like than IL-6, I'd say barely, not a lot. I mean, and again, just what I'm ultimately going to say, in a way, is that maybe these small differences, if one of the small differences here is interferon gamma, well, that's huge. You say, is IL-27 more Th1 than IL-6? And you'd say, yeah, you betcha. It regulates interferon gamma. But if you look at it this way, you'd say, not so much. And so I think both of those answers are true, and it just, again, reflects our ability to sort of think about this. So what are the mechanistic underpinnings of this? Again, I'm running out of time. I'll try to be quick. If you look at the transcriptomes that are induced in wild-type cells, and then you look at cells that lack STAT3, you can see that the transcriptomes are entirely dependent upon STAT3. And that was a surprise to us, that we would have thought that IL-27, that STAT1 would have a more profound effect on IL-27 signaling. In fact, when you remove STAT1, you have this very strange effect that you now allow both cytokines to access STAT3 more effectively. So you actually change the transcriptome by the loss of STAT1. But conversely, if you were fortunate again at the NIH have this nice ability to do bench to bedside, so this is a disease called chronic mucocutaneous candidiasis, which is due to STAT1 mutation. And we have these patients, thanks to Steve Holland at the NIH. And we wanted to look at the effect of STAT1 gain of function on IL-6 and IL-27 transcriptomes. And that's shown here. And you can see that ordinarily, as I alluded to before, that if you take, take cells and stimulate them with IL-6 and IL-27, they're way more similar than we would have predicted. But what happens is in the gain of function patients, you have more now diversity. STAT1, increased activity of STAT1 makes the cytokines seem more different. And that's quantitated here. So let me just summarize that and just say that there's way more overlap in the transcriptome that we would have predicted. And this is very interesting because my colleague Bill Paul, who I'll mention again at the end of the talk, noticed this in 1989 and noticed this sort of an oddity that when we were first thinking about cytokines, we expected them to have very specific functions, and in fact, they disturbingly had this tremendous amount of overlap. And so the TH-ness, we would say, of cytokines can be really overstated, but it could very well be that it's the small differences that matter. And so when you have this whole transcriptome, ironically, what's important is to go back and understand exactly how that cytokine regulates this particular gene. And as I'll show you in a moment, the good news is now we can do that. The transcriptional output is driven by STAT3, whereas the ability to access STAT1 is important for specificity, and that's really revealed in these patients with STAT1 gain of function. And that's exciting because STAT1 and STAT3 levels are very variable. And again, it's not something that people thought about in the early days of the JAK-STAT pathway, but now people like Chris Byron think about this a lot, and George Stark. So again, this is in cartoon form what I just told you here, that STAT3 really regulates the total transcriptomic output, whereas STAT1 really regulates the specificity. Okay, so the next step in this project sort of happened in 2007, when after the genome was completed, and when we got these machines, Illumina machines, to do next generation sequencing. So now here you could return to this problem again and say, okay, how does a cytokine impact a cell transcriptome? That's straightforward. But where is a transcription factor like STAT3 going in the cell to mediate that? And the answer, in one sense, this is cells stimulated under Th17 conditions, looking at STAT3, and you can see STAT3 binding to the promoter of the IL-17 gene, STAT3 binding to the promoter of the IL-17F gene, and again, STAT3 binding to the T-cell specific transcript of RR gamma C, the master regulator of Th17 cells. So again, all of that was, to me, it was just tremendously exciting. I couldn't believe it that in my lifetime I'd be able to say, well, look at the entire genome and find out where a STAT goes. So that was exciting in and of itself, focusing on the genes. But as I'll get to, you also had this other stuff, like, oh, well, you know, STAT3 is binding here. And some people at the time said, nah, Chipsy, you know, STATs bind everywhere in the genome, all these transcription factors. What do they do? How important is it? I'll come back to that in a second. So we identified at that time thousands of STAT target genes in Th17 cells and Th1, Th2, et cetera. The targets were all the things that you were really hoping for, products of Th17 cells, and we were able to, in that case, find a direct link between the STAT, the extracellular signal, and then what genes were being transcribed. But you also saw this, lots of binding other places in the genome. And the other thing that you could do, start to do at this time, was to do ChIP-seq with things like, with histone modifications. And what you see is that you see modifications of histones where there's accessibility of loci at the same places that STATs are binding in this case. So that was the other big surprise when we got the genome, is that the genome was mostly not genes. And you know, at the time, it was a little bit disturbing that only 2% of the genome is genes. And then you'd say, what is the rest of this stuff? And at the time, people said, you know, this is just genomic junk. I would say that it's still a bit of an argument. There are people that argue for the junk hypothesis. It turns out that we're not the biggest genome. I think onions are way bigger than our genome. It's cool, right? Because onions have so many layers, right? You know, it's like they're complex. We should have known that. But I think the lungfish, I may be wrong. This is being recorded, so I should be careful, make sure what I'm saying is true. But I think lungfish have the biggest genome of all. So that's really disturbing that, you know, that all of the complexity of us, if you want to ascribe it to genes, it's a little disturbing. That's only 2% of the genome. But initiatives like ENCODE have shown that 80% of the genome is active, including a lot of the genome is transcribed in areas of places where they're not conventional genes. And most of the genetic risk of disease is actually in the junk, not in the genes themselves. So again, we were very excited about this, and we sort of were thinking about what does the junk tell you about, again, this basic problem that I was thinking about, you know, way back when Marty Rodbell was telling me about signal transduction. So the other thing that was discovered a little bit later is that you can define what seems to be an enhancer by this signature, and that's histone 3 lysine 4 monomethylation is a mark of poised enhancers. And then if you have histone 3 lysine 27 acetylation induced by enzymes like P300, and you can actually have transcription of a different type of RNA, an enhancer RNA, that's the mark of an active enhancer. So we became very intrigued by this, and Golnaz Vahedi in my lab decided to enumerate this and came up with this very surprising result, or at least surprising to me, which is that Th1 and Th2 cells, which started out a few days before in vitro, had 20,000 or so genomic switches, but only half were similar between Th1 and Th2 cells. So you had 10,000 separate switches in Th1 and Th2 cells, and basically no switches that were shared between Th1, Th2 cells, macrophage, and embryonic stem cells. Now, again, as luck would have it, in that fall in the same issue of Cell, others were doing similar things and came up with pretty much the same analysis, that you had this tremendous diversity of switches, and also that the major role of the environmental sensors, in this case the STATs, were really the thing that were driving the enhancer landscape. And the lineage-defining transcription factors had less of an impact. So I think that the prevailing wisdom prior to this was that if you're a lineage-defining transcription factor, you should really supervise the epigenome. But in fact, that really is not the case for some of the things that we call master regulators in T cells. And FOXP3 was a good example of that. Sasha Rudensky showed that the FOXP3 comes in after the genome is modified. So the other thing that we've learned just very recently here is the distribution of enhancers in the genome is very asymmetric. And this is the work of Rick Young and my boss, Francis Collins, and we had a paper earlier this year. And what you see is if you look at binding of mediator K27 or P300, you see this very asymmetric distribution of enhancers. Now, to me, I grew up in New York, as you heard. This wasn't so surprising to me because I knew that Manhattan had these big skyscrapers, and then I went to college in upstate New York, and there were no skyscrapers, so I get that. This is a little port of Manhattan. It's this dense, you know, where a lot of activity is. So to me, this seemed like it made a lot of sense. Now, I should say I wrote a review, and I used that metaphor, and I think they didn't like it, but I like it. You tell me whether you like it. Now, again, this region also here is enriched for GWAS hits. And the proposal had been made by Rick Young that the appearance of super enhancers was linked to the idea of cell identity. And so we really, in Golnaz's paper here, we were very interested in understanding the most relevant T-cell switches, because our thought was this is really telling you, well, you know, what are the Manhattans for T cells? So for any given cell, Manhattan will be in a different place, but, you know, what you're looking for is you're looking for the skyscrapers. And so our idea was that this really was a non-biased way of identifying cytokine-dependent regulatory nodes. If, again, this whole idea, if you're having a signal outside the cell, what is it telling a cell to do, how does it do it, and where is that action? So, again, we reanalyzed Rick Young's data in this paper, and we found, just like Rick Young found, is that in embryonic stem cells, the part of the genome that has super-enhancer architecture are transcriptional repressors. And that makes sense because embryonic stem cells are mostly about what they shouldn't be doing. In macrophages and in T cells, the regions that have super-enhancer structure are chemokine receptors, and cytokine receptors. So on one level, this was very exciting. On the other hand, you know, if you're an immunologist, you sort of say, really? What are you expecting? Of course, T cells are regulating cytokines. That's a very expensive way to figure out what you already knew. The paper's published, so I'm not going to go into a lot of the detail of this. We did have a big surprise, which was the super enhancer is a gene called BOC2, which fortunately we had just written a paper in Nature a couple of years ago on how BOC2 was this critical regulator of tolerance in T cells. And in all subsets of T cells, the thing that scores the highest super enhancer structure is BOC2. So there were some obvious things and then some less obvious things. And the other thing that's less obvious is that long non-coding RNAs are enriched for the super enhancer architecture. But this part, at least that part, makes sense. I like things when they make sense too. So the other thing that we found was sort of a reiteration of Rick Young's data, is that if you look for loci that were associated with autoimmune disease, they're enriched for super enhancer architecture in T cells. And so you could see examples where you have this enrichment of super enhancer architecture. And a nice comparison is type 1 and type 2 diabetes. Here you have an enrichment of T cell super enhancer, and here in type 2 diabetes, a disease you don't usually think of as having a heavy immune component, you don't have this tremendous enrichment. So the other thing that we were excited to see was that if you treated cells with Tofacitinib, you could see that Tofacitinib is primarily acting on loci that have super enhancer architecture compared to genes that have typical enhancer architecture in these violin plots. You see these very dynamic changes here. Okay, so to summarize this part, you have dynamic tissue selective expression, and that correlates with super enhancer architecture. Cytokine and cytokine receptors are the dominant class, but BOC2, what we've referred to as a guardian transcription factor, is really the champion, and so it really does have this ability to supervise the genome and prevent aberrant transit into effector cells. Polymorphisms associated with immune-mediated disease are more likely to have super enhancer architecture than typical enhancer architecture, and rheumatoid arthritis-associated genes with super enhancer architecture are preferentially affected by JAK inhibitors. So I'm running out of time. But I just wanted to summarize a few things. So membrane nucleus signaling has gotten very interesting since the 1980s. I mean, Marty Rodbell's discoveries in his class was amazingly exciting, just linking the biochemistry, but we've made enormous advances in taking the first steps in the plasma membrane to getting it all the way to the nucleus. And we've learned a lot about the elements. So this is a very exciting basic advance, but as I showed you, it also provided lots of opportunities for new therapies. So I think, as I was talking to the students earlier, I was thinking the challenge, of course, ahead, in a way, is now we can measure everything, right? We can measure transcriptomes, we can measure epigenomes, we can measure changes in the proteome, et cetera. And so I now think that the real challenge is integration of signals that impact chromatin organization in real time. So we should be able to do this. Most of the things I told you about were snapshots. So increasingly we'll be able to do these things on single cells. We'll be able to do them over time. And really, this is what I would like to understand, is how do extrinsic and intrinsic signals really modify the chromatin architecture to enable transcription. And things I don't have time to tell you. We're now beginning this with techniques like ChIA-PET, et cetera, et cetera. But the exciting thing is we have great new tools like CRISPR-Cas, super resolution microscopy. I think we're really on an exciting new sort of place for really now understanding signaling in a way that we couldn't possibly dream of. And it should be a lot of fun to do this. I'm really looking forward to the next 20 some odd years of studying signaling. I'll stop right there. I did want to acknowledge Bill Paul. Bill Paul has been a tremendous mentor to me over the years. Sadly, if you know, Bill died earlier this month, and was just this terrific person who really pushed immunology both at the NIH and around the world, and I wanted to sort of honor him. Lots of other people to thank along the way. Lots of people in my lab, I think I mentioned them. Also, our collaboration with Francis Collins on super enhancers, I mentioned. But again, NIH, an amazing place where people are extraordinary collaborators and willing to sort of share their expertise. And then long, very satisfying collaboration with Pfizer, especially Paul Chingalian, and now more recently with Jim Clark and J.B. Tellier. So thanks very much. I think I've run out of time. Thanks.